Welcome to Imprint, an analogous video game that was developed to aid your understanding of the epigenetic phenomenon of genomic imprinting and extensive research that has contributed to our understanding of it. We know that organisms inherit two copies of their genes, one from their mother and the other from their father. However, the phenotypic expression of these genes is predetermined by the parental origin and expression pattern. This is known as genomic imprinting, where one of the homologous parental alleles is epigenetically marked via DNA methylation during gametogenesis. Consequently, the appropriate imprinting of parental alleles is necessary for a balance of gene dosage to be established. This in turn allows for the regulation of normal embryonic growth and development of specific lineages. Therefore, when this balance is disrupted, the resulting effects of these imprinted genes are expressed in the organism's phenotype. But how so? For instance, if either paternal or maternal genomes are duplicated, a surplus or deficit of gene dosage and its corresponding phenotypic effects will ensue. Here we can see Sheila Barton and her team's gameplay on the androgenic route that investigates androgenetic embryos which only contain two paternally inherited genomes. While navigating the game, Sheila's team was approached by a merchant to complete side quests. This involved obtaining fertilized one-cell eggs by mating the F1 female and 129HD male mice from the merchant, culturing them to the advanced blastocyst stage, isolating the inner cell mass via immunosurgery, then injecting them into host blastocysts from the merchant's CFLP mice. Upon completion, the team unlocked the Chimera Creator achievement and passed the checkpoint. As they progressed towards the final battle against the boss, they collected tokens on the way. However, unbeknownst to them, the AG tokens are representative of androgenetic cells and the status bar fills correspondingly to indicate the embryonic composition of AG cells. So with a 50% reading, oh no, the boss was the chimera that they had helped the merchant create. It was far more formidable than predicted. Its size, weight, and anterior-posterior axial length were increased, and it had severe skeletal abnormalities, evident in its disorganized and twisted ribs, severely scoliotic spine, and thick contorted tail. But how? Well, AG cells undergo extensive cell differentiation in the presence of normal cells in chimeras, significantly contributing to phenotypic alterations during embryonic development by proportionately increasing embryonic size and enhancing embryonic growth. This is evident in the increase in weight from 92 mg to 138 mg and an anterior-posterior axial length of 8.9 mm to 10.3 mm, corresponding to the increased AG cell contribution from 0% to 50% that Sheila's team observed. However, a contribution of AG cells greater than 50% was invariably lethal and further observations could not be made. These alterations are attributed to the overexpression of the insulin-like growth factor 2 and the absence of this receptor due to imprinting. This is because AG cells contribute scarcely to the brain, but preferentially to mesodermally derived tissues such as the heart and skeletal muscle, the very tissues in which the IGF2 gene is expressed. Furthermore, regulation of IGF2 levels via removal of excess ligands is hindered in the absence of IGF2R, contributing to the growth-enhancing effect of AG cells. This contrasts the other route that investigates phenotypic effects on gynogenomes and parthenogenomes, or simply put, embryos that only inherited two maternal genomes. The presence of GG or PG cells in chimeras resulted in growth retardation and a proportionate reduction in embryonic size, evident in a weight of only 50 mg and an anterior-posterior axis of 7.6 mm. Therefore, these embryos are phenotypically reciprocal to the AG embryos both in size and the distribution of these maternal cells, contributing to the brain but negligibly to the heart and skeletal muscles. This is due to production deficiency of IGF2 ligands coupled with greater ligand removal due to excess IGF2R. This demonstrates that the co-expression of paternally expressed IGF2 gene on distal chromosome 7 and maternally expressed IGF2R gene on proximal chromosome 11 regulates normal embryonic growth by respectively enhancing and suppressing it. Therefore, parental genomes are functionally non-equivalent during development, and both a paternal and maternal set are necessary for normal development. Sheila's team was prepared for Imprint after analyzing previous players' gameplays and were aware of the aggregation technique that the players had used for the Chimera quest. Consequently, they decided on completing it with the injection technique, a novel, never-done-before technique that improved development efficiency of the chimeras, paving the way for a new tactic in Imprint that others were able to use to better establish the paternal genome's role during embryonic development. Not only did Sheila's team's similar results validate findings from previous players using AG embryonic stem cells instead of AG inner cell masses, they managed to unlock the achievement Eureka with their discovery on embryonic growth effects, as it had not been previously reported before. Hence, both the team's corroborating and novel findings allowed for further identifications of more than 100 imprinted genes at present. This strongly supported and resulted in the currently widespread acceptance of the parental conflict theory of Hagen-Westobie that was proposed in 1989, predicting the involvement of maternally expressed genes in resource conservation from the fetus and paternally expressed genes in resource extraction to the fetus. This was Imprint. Would you like to play again?